Welcome back. The topic is your keeping track of moving cells, pet maps, and the cell lineages. Indeed, one of the most important conclusions of the developmental anatomist is that the embryonic cell do not remain in one place, nor do they keep the same shape. Very interesting thing. Indeed, one of the most important conclusions of the developmental anatomist is that embryonic cell embryonic cell do not remain in one place, that is your epidermal cell, nor do they keep the same shape, that is your mesenchymal cell. There are two major types of cells in the embryo, where the epithelial cell, which are tightly connected to one another in a sheath or tubes, where your mesenchymal cell, which are unconnected or loosely connected to one another and can operate as an independent unit. So within these two types of arrangement, the morphogenesis is brought about through a limited repertoire of variation in cellular processes, duration and the number of cell division, cell shape changes, cell migration, then your cell growth, cell death, change in the composition of the cell membrane, etc. So from the beginning, direction and the number of cell division. Just think about, if the direction changes, then it's like your forelimb will moving in a different direction of your left forelimb, whether your right forelimb moving in the other direction. So this kind of problem, and the number of cell division, and I told you, multiple, if every single cell in your body multiplied, then will be enormous and very difficult. Just something. Okay, the cells shape changes, cell migration, etc. So the fate maps, that's what I'm talking, the fate maps. It's not like that your geographical map, rather is the lineage of maps. That means one cell which divides another cell, then another cell. So the way it's produced differentiated tissues, organs, so that map is your called fate map, beginning from the zygote to the blastomeristate cell, then your different germ layer cell in gastrula, followed by different organs, tissues, organelles, etc. Organ system, not organelles. The fat mass of the vertebrate at the early gastrula stage. Here is shown the cells of these that will form notochord occupy at the central dorsal position. The notochord the one which produces notochord, here it is. In the central, the neural ectoderm is surrounded by a less dorsal ectoderm. This is a neural ectoderm, less dorsal region, which will form epidermis in the skin and indicate that the anterior end of the embryo, the P in the posterior end. So different fates are being shown here. A is anterior, P is your posterior. So on the top you can see towards the anterior is your epidermal ectoderm, followed by neural ectoderm. You see it is the ectoderm. Then your mesoderm, which includes your derivatives of your which includes your mesodermal cells, endoderm, etc. So dye marking, here is shown here, and yellow color is applied to a cell, which on further differentiation will lead to a particular region being colored, and by this we can give an idea, or we have an idea how the cells of a particular region can differentiate, and located anywhere else can be determined by using these dyes, and that will be helpful to determine this fate map of that particular organism, a particular region, that cell type, etc. Here also some done. Here are agar chips with dye is being emerged with this that embryo, which leads to further embryonic developmental stages, that leads to the cells in that region with that agar chips dye, leads to differentiator, and that can be written, and then later on it can be observed by dissecting. You can see here is a pattern showing how these organs, those mesenchymal cells are moving, while epidermal cells are moving. So this movement and localization of these particular cells in the embryo can be determined, which is helpful to determine these embryonic fat maps. Fluorescent dyes, the fluorescent conjugated dextran being used here can be injected into a single cell of any embryo, and the descendant of these cells can be seen by examining the embryonic under ultraviolet lights. So the same thing is here, as the dye you have inserted, because during the cell cycle what exactly happens, the cytoplasmic substances get multiplied and separated by a membrane, and this way the dye you have emerged in a single cell will lead to distributed throughout every single cell that being emerged from that particular cells that you have taken to insert your dye. So in this case you can see here in the embryonic stage a particular region with some cells are injected with some fluorescent dye, which is activated by some laser treatment, and that further cell division during this embryonic growth can be observed by using ultraviolet light to show the fluorescence property of that region. The region where showing this fluorescence light coming out can be taken as a PET map to observe the movement of that cell from that region to these organelles, or we can say that the differentiation of the cells occur to form that organ from that particular region of this cell. And one more thing, another thing is very important thing. This is a transgenic DNA chimeric, where the different you better see in this region here a quill embryo of that particular time hours in that particular region has taken out and been inserted in a chick embryo, and this gives this kind of phenotypic expression. That means this region of the cells determine this phenotypic region of that organism. So this is how also we can know the fat maps of a particular tissue. One of the most interesting things is the green fluorescence protein. This is a gene which code for the protein that what we call green fluorescence protein, and the fluorescence protein because it fluoresces in a particular wavelength of light, and it's and by this green fluorescence protein we can observe the where a protein is which region is localized this protein, and by that we can observe the fat map of that organism. And two things that I want to tell you that this is green fluorescence protein. Another one is the luciferase protein. The two protein genes are being used to different aspects or different requirement. The luciferase gene is being used to localize to not localize, sorry. The luciferase gene is used to show whether your desired mRNA desired protein is produced or not. Whereas your green fluorescence protein, when it hybridized with a promoter, it will show the localization of that protein inside the body. And here is shown some green fluorescence protein being tagged with that particular region of the cells with gene. So each and every single cell dividing with that particular gene in its green fluorescence protein also get replicated, and that's showing this region of expression during the developmental stages. Here it shows the somite region which is being tagged with the green fluorescence protein gene. It's showing the neural cell and some neural tube region, showing that it emerges from that region of the cell of somite cell. That means here is written a chick embryo containing An active gene for the green fluorescence protein expresses the green fluorescence protein in every cell. The brain is forming on the left side of the embryo. Yes. Mm, and the bulges on the forelimb are containing this head ectoderm and initiate the eye formation. The region of the neural tube and neural crest is the presumptive neck region is excised and transplanted into a similar position on level wild type of embryo. One can see it by green fluorescence protein. That's what I'm trying to tell. This you can observe by this green fluorescence protein. Then later on, a day later, one can see the neural crest cells migrate from the neural tube to the stomach region. And then in more 4 days, the neural crest cells have spread out in the gut and esophagus to the anterior end of the hindgut. So this movement of this particular cell are differentiated into different kind of regional organs can be observed by this green fluorescence protein tagging. So this is the way we can also determine the fat maps of that particular organism, of that particular cells. That's it. Thank you. It's almost done, finished. Thank you.